Hello, everyone. Thanks for joining. Yeah, Tutor Glendon here. Just checking, can you all hear me? Okay, sound check, sound check. Can you all hear whatever I'm saying? Yes, yes. Okay, good. Yeah. Okay, if you all see the screen right now, I believe you can see the screen and hold on. Yeah. So can I get you to go to the Zoom chat? Now the Zoom chat, I've placed the link there and there will be today's lesson materials. Okay, so the lesson materials are all online for today. Can I invite you all to go to the Zoom link, okay, and download it? Okay, sorry, the link in the Zoom chat to download it. We were using that for lesson today. All right. Once done, just put a thumbs up beside your name. Or if you have any difficulties, let me know. Then I can help you download it. All right. So go to the Zoom chat and download the link from today's materials from the link. Awesome. Good job, WY and Amelia. Yeah, we'll just wait for another 8 minutes. In the meanwhile, you can chill. Once we start, I'll let you all know. Okay. Hi Annie, welcome. Do take note the instructions. So I've left the material for today's lesson in the Zoom chat. So do head there to find the link to download it for the lesson. All right. If you need any help, let me know. But once you are done downloading, you can just give me a thumbs up. Okay. Hi Kimpo. Yeah, thanks for joining us. So do look at the instructions. Head down to the Zoom chat. There's a link there for you to download the lesson material for today. Just scroll all the way up. Okay, so download the PDF document. That's all the lessons materials you'll be using for today. So once you're done, just put a thumbs up in the chat, all right, to let me know. And we should be starting really quickly at now 3 minutes. Okay, so maybe if I can invite you all here, okay, all five of you, can you maybe type in the chat what are some issues you have with the topic for today, like cell structure as well as movement of substances, anything that you would like me to help you with, so that later when I do the session, it can be more targeted. Hello, people who just joined us. Right? Hi there, people who just joined us. Welcome to the session today for Pure Bio. The learning materials for today's lesson can be found in the Zoom chat. So please go ahead and download it. In the Zoom chat, there is a link to today's lesson. So please go and download it before I start. Good thing, okay, the Zoom link, you can actually just scroll upwards. You will find it there, but don't worry, I can repost it again. Just scroll upwards. Hi, Tabitha. Thanks for joining us. Do find today's lesson materials in the Zoom chat. Just scroll all the way up. You will see a link and you can download it. Oh, you all can't scroll. Okay, lah, I keep posting. No worries. That's strange. I have enabled. Continuous meeting. No worries. I'll just keep posting. Hi, those that just joined us, do check the Zoom chat for the link to download today's lessons materials. I think some people are still joining, so let's just wait for another 4 more minutes. Those who are unable to get the link or you don't see the link in the chat, just drop me a text on the Zoom chat and I'll gladly help you. All right, I'll gladly help you. Don't suffer in silence. Those that just joined, hi, welcome. Those that just joined, thank you so much for joining us. Do go to the Zoom chat to download today's lesson materials. Yeah. Yes, I think we might or might not. Well, I'll be recording a lesson regardless, JL. So yeah, I'll be recording a lesson regardless. Hi everyone, just joined brand new. Do check out the Zoom link. There is the link, the Zoom chat, sorry. There's a link there for you to download today's lesson materials. And just to be clear, this is the Tech 3 Pure Bio session. If you're here for IP3 revision, that's at 7 p.m. No, 7:30 p.m. So do take note, that's at 6:30 p.m. No, 7:30 p.m. Yeah. So that is the latest session. So this is the Sec 3 Pure Bio session. If you're IP student, please go for the second one. All right. Hi, Zena. Nice to see you there. Okay, please find today's lesson materials in the Zoom chat. Okay, and please download it. More people are slowly streaming in. 18 people, not bad. Okay, another 1 minute's time, I'm gonna start. So once again, gentle reminder, if you just joined us late, the Zoom chat has a link there to download today's lesson materials. So everything's all online. So do give it a download, right? And I'll be using that for today's lesson. And at the same time, feel free throughout the entire lesson to unmute yourself, okay, to ask some questions. I know you all won't, okay, but feel free to do that. Or at the same time, you can drop some questions in the Zoom chat as I'm going through the topic for today. Right? Okay, if not, now yours, let's start the lesson. Okay, let's get your time here worth it. So before I start, a short introduction about myself. Who is this gentleman here? Okay, talking and why is he teaching bio? So just a very short introduction about myself. Okay. Before that, let me check Zoom. Which screen are you showing right now? All right. Testing. Hey, yeah. Hi, everyone. Can you see the screen? Should be this first page. So yes, welcome to the WA1 Pure Bio Revision session. Okay. And we are overmarked. So generally, we have taught quite a few students, about 5,000 students across the last few years, and we have a few different branches. Okay. Now, those who are in combined bio, you're perfectly fine. In fact, both topics are very similar. Later, I'll tell you which parts you can ignore. So, but this recap will serve to be a very good help for both the pure bio and the combined bio people. So don't worry about that. Okay, you are perfectly in the right place. Now, yeah, we have 7 different branches. We have Woodlands, Bishan, Jurong East, Bukit Timah, Marine Parade, Tampines, and Kovan. Hopefully, there's one near you if you're interested to see what our actual lessons are like in person. And across Telegram, now, pro tip, if you are not in our Telegram channels right now, you are doing yourself a disfavor. Why? Our Telegram channels have a lot of free stuff, a lot of good learning materials, a lot of hacks on how to study better. So if you're not inside it right now, what are you waiting for? Okay, go and find out how to join our Telegram channels. Okay, now, uh, who am I? Okay, I'm Tutor Glendon. Nice to meet all of you. And the first thing you notice is, wow, this guy teaches A-level, O-level, IP bio. Is he the right person for me? That is just there to share with you that I love biology. I've been teaching biology for many years and I've taught many different levels. So feel free to ask me whatever questions you have. All right? Feel free to ask me whatever questions you have. I'm formerly from MOE. I used to teach, okay, in the MOE school for about 7 years, and I left to do private tuition for the last 3 years lah. All right? And people ask, hey, how good are you as a teacher? All in all, generally, my students do pretty well. Okay, some of them are here today. And about 70% of my students achieve distinctions. It's purely because they do the hard work and they hustle based on my guidance. Now, there are 2 links in front here. To those of you who just joined me, hi, welcome. Do head to the Zoom chat. In the Zoom chat, there is a link for you to download today's lesson materials. Okay, once again, do head to the Zoom chat to download today's lesson materials. Now, fun fact about me, okay, now there are these 2 ladies here, which I don't think any of you will know. You probably might know the lady on the right, who is IU, okay? Lady on the left, maybe a bit too old for you. But why am I putting these 2 people here? These 2 ladies are not my girlfriends. They are not.
Hello.
Hello, May. I love the excitement. I can hear you so
Oh my
loud.
god.
Yes, you're either a fan of IU or Miss A from Suzy. But these 2 ladies here are not my girlfriends. Generally speaking, I wish they were. Don't tell my wife, okay, she's next door. I'm having this lesson at home. But I actually use these 2 ladies to teach biology. I kid you not, okay? But it's mostly at the A levels. I use these 2 ladies to teach the concept of respiration, okay? Using a story of how I'm going to marry either of them. So if you're very interested in what nonsense is this, don't worry. Today's lessons will be all such stuff using very funny stories to help you learn biology. Okay? Now, besides that, I think I'm pretty decent as a bio teacher. So my last year cohort, okay, these are some of the grades that people got, mostly A1 and A2. Okay, I'm going to mute Xander. All right, I'm going to mute. Okay, I can't mute Xander. Oh no. All right, so generally, I think I do pretty all right lah. These are some testimonials from the last year's A-level students. Okay, now, these are some online reviews from the students who have taught me. So yeah, so you are in good hands for today's review session at the very least. More fun facts about me. I love to cook. So the thing on the right there is something I cooked during COVID. And last year in Christmas, I cooked a large family dinner. I see biopractical kind of like cooking. So to me, it comes hand in hand. All right. That's me and my young son. Okay. Now he's 8 months old. He's really cute. So this whole picture is just for me to be a proud dad. Then some other fun fact about me, I used to be a Team Singapore athlete. I used to play floorball. So this is at the 2019 SEA Games, where I last represented Singapore. I retired so that I could have this little fella on the left. And then lastly, this one you will never be able to find if you do our bio coffee. So a long while back, my wife and I, now that's our hands over here. We are not hand models, but we were featured in a Lego commercial in Singapore on YouTube. As you can tell, the middle thing here is Marina Bay Sands. So when they were launching it, they wanted us to appear in their advertisement. Because me being a romantic, right? Okay, Mr. Paul, very romantic one. I proposed to my wife in front of Marina Bay Sands. So they were like, we want your story. So I gave the story. So you can try and find this. It's very hard. If you are in my class, okay, and one day I'm nice, I probably will show it to you lah. Okay, now that's fun facts. Now is the part where I tell you, don't worry, whatever stuff I say about bio can be trusted. Okay, because why? Firstly, I did a degree in biology. Then I got a teaching scholarship to be a MOE teacher. That's my mom. Okay. Then there was me at my graduation giving a speech. Long story short, was pretty funny. People enjoyed it lah. I hope they had fun. Then there is me teaching in school. Yes, that is a strawberry suit. Yes, I taught a class in a strawberry suit. Why? Why not? Because life is too boring in school. Let's make it more interesting. Okay, so I'm very confident that day. Everyone remembers the lesson, but no one remember what I taught. Okay, but that's me teaching bio. Then I left teaching to do two other boring jobs. But right now in OVERMUGGED, this is what I mainly do. Okay, besides being a bio teacher, I'm the subject head for bio for JC, IP, and secondary. I also train these little fellas. So I train the teachers to be better teachers. All right. And on the note of subject head biology, so I don't know whether last year you all were following us during the O-level period, but we had practicals sessions to train your Sec 4 seniors. So for bio, I had to prepare the Sec 4 practical. And the practical that I created for everyone to practice was potato. Guess what came out for O-levels? Okay, almost the exact same thing. So in the words of my ex-student, okay, he was like, Mr. Pua, do you find out? Do you know what happened? I was like, what happened? Your practical came out. I was like, wow, typo. Okay, so what I'm trying to tell you here is not that I'm super good. Okay, no, that was pure luck. But what I'm trying to tell you here is I'm a teacher who's able to analyze trends and see what I can do to best prepare you for, okay, your assessments. So that's what I'll be doing later for your WA1. All right. And these are my three teaching philosophies. Actually, in all honesty, this page is just for me to show you my son because he's very cute. Okay. He's smiling because just like me, he believes, okay, his life is a performance. Every day he's crying. Right now, you can hear him crying. Okay. Teaching is a performance to me. He's laughing because I make him laugh every day. Same thing. I think education is best done through humor. And he's a baby. I cannot give him complex instructions. I can only say the simpler stuff like eat, sleep, and poop lah. So it's my job in teaching to make the complex simple. All right. Okay. Now that's all about me. Now moving a bit to the company. Some fun facts. So at last year's A-level, O-level results, okay, about 70% of our students achieve distinction. Our tutors are from reputable universities in Singapore. Okay, and the teachers are mostly from MOE as well as Learning Lab. Okay, MOE as well as Learning Lab. And if you come to our classrooms, you realize, wow, there's quite a lot of vibrant learning environment, a lot of free educational resources, nice food, chill place, good place to relax. Okay, in fact, right now you have a problem. Our students tend to study at our study areas a bit too long, but it's okay. It's a good first world problem. To those that just joined us, hi, welcome. I'm in the middle of my introduction. For the learning resources for today, please head to the Zoom chat and you will find a link that I've been spamming. Do download the learning resources from that today. Okay. Now, what I'm giving you is free resources, and I think throughout the whole entire Singapore system, people kind of already know OVERMUGGED for free stuff. So we are okay giving free stuff because it helps you. So on top of this free thing that I'm giving you right now, we also have other stuff which are even better. So if you're our own student, you will benefit from curated notes, weighted assessment revisions. So the ones that you're having right now is the free one. The one that my students will be going through, different level. They're going through an actual WA paper. Okay. Then we have mock exam papers, prelim papers, science practical notes, practical, which I mentioned just now, cheat sheets, and after class consultations. Okay, so a lot of good stuff, lots of good stuff if you join us. Okay, this one later I'll come to that. All right, so my job here is not to make you join us. My job here today is to help you ace your WA1. So let's get straight to that, shall we? So let's go to this part over here. Okay, so can you please thumbs up for me if you have downloaded my learning resources and you are ready to learn about cell structure and organization? Thumbs up for me in the chat if you are ready. Awesome. Thank you so much. Okay. So in honesty, here's what's going to happen. I'll be going through this topic first. Then we'll go through some questions. Then I'll go through transport of movement or transport of substances and going through some questions. I will not be reteaching the entire topic. But what I'll be doing here is to double down on areas which you might find confusing. At the same time, is for me to pass you knowledge, okay, on things that you might find hard to remember. Right? Now, cell structure and organization. Wow, this one, Mr. Pua, super hard to remember. Okay, it's like just pure bio memory work. It's everything I hear about bio. I'm going to consider a fact. It is true. It is true. It's a lot of memory work for this topic. Okay, but I think it's always easy to remember something if it's more interesting. All right. Now, the way I like to approach this topic is this: cell structure and organization is simply a Taylor Swift concert. Oh, wait, what? Did you just say Taylor Swift concert? Yeah. Cell structure and organization is like the Eras Tour concert that Taylor Swift just did last year. And I'm going to convince you of that in a very short while. All right. Now, this portion here on the cell theory, okay, I won't be reteaching everything, okay, but I just need you to know. In your school paper, in the WA, at max, they will test you only 1 mark worth of this. Why? There are many other things for them to test you. Then, it's so hard to remember, Mr. Phua. Let me help you with that. The answer for cell theory, if you're ever lost in cell theory, is you stop doing your paper, look at your hand, look at yourself. You are the answer on remembering this. Because why? Last I checked, I'm a living person. I hope you are a living person. I hope there's no AI person here. If you're living, what are we made up of? Cells, right? If I cut now, got red blood cell drop out. If I scratch my skin really hard, I get dead skin cells. If I pluck my hair, I get a hair follicle, which is a hair cell. So that gives us the very first thing about cell theory. I'm living and I'm made up of 1 or more cells. So no longer need to memorize it. This is just understanding. You are the answer to cell theory. If they ask you for 1 mark, what is cell theory? Think of yourself. I am a living thing. I'm made up of 1 or more cells. Okay, now I am more than 1 cell. Where is the 1 cell that is living? This is bacteria. Okay, then I'm going to help you remember this with the third point. All right, there are so many here, a bit hard to remember. I'll use the third point to help you remember this because why? All cells are formed from the division of pre-existing cells. Wow, very, very cheap. Okay, now basically, this is what you learn in lower sex science. How did I get my little baby boy? Sperm meet egg fertilization. When that 1 egg has fertilized with the sperm and it becomes the original 1 cell, how does it go from being 1 cell to my cute little chubby chicks baby? Oh yeah, so that 1 cell divide and divide and divide. So that's how you know this. Okay? All cells are formed by the division of pre-existing cells. From the sperm and egg fertilizing, basically you are how you were created. How you existed, sperm and egg from your parents fused together, and there was a lot of cell division. So all cells are formed only by division of pre-existing cells. And with 2, there's more than enough. Okay, I kid you not. Because no school paper is likely to test you 3 marks. There are so many other things to test you. Okay, so for cell theory, this is my advice. Use yourself as the answer to remember. I'm a living thing. I'm made up of more than 1 cell. Okay, so all living things are made up of 1 cell. I came from a sperm and an egg, fertilizing together. From that first cell, divide, divide, divide. So all cells came about from the division of pre-existing cells. That is how it's easier to remember this part over here. Okay. Now, microscopy part, this one, okay, I just need you to know there are 2 types of microscopes. If the question comes out, okay, likely it's going to be a light microscope. Okay, it's going to be a light microscope. Why? Electron microscope, you are never going to touch it until you go to university. In A-levels, you learn a bit, but O-levels, they tend to like to test light microscope more. Okay, so for WA, maybe can focus on that, right? Now, for light microscope, basically, it's not the most powerful microscope. So what can I see? Very simple, straightforward. If I ask you to draw for me a typical animal cell now, okay, you draw for me. What will you do? Oh, cell, rough shape like this, or then got nucleus. That's all. That is all you can see in a light microscope. So if you're lost in the exam, microscope, light microscope, what can I see? Just draw what you can draw. You can draw. Okay, what you can draw as a normal non-artistic person is all that you can see. And that is the tip for the light microscope. Okay, you will only see the nucleus, cell membrane, and the cytoplasm. Where's the cytoplasm? Yeah lah, the red color part. All right, so you will only be able to see these 3 things that you can draw. Okay, in an animal cell. In a plant cell, okay, now, what will I also see? Where is the cell wall in a plant cell? It is the outermost thing on the outside. Okay, outermost thing on the outside. Sorry, a bit hard to draw on my stylus. So you will definitely see cell wall and cell membrane. Okay, now erase this. In the plant cell, okay, you will also see that big middle thing that looks like a big water bag. What is that big middle thing in the middle? Your large central vacuole. So the nucleus will be here, all right? Then you will have some green color chlorophyll, all right? Now in the exam, how to remember? You can see chlorophyll. Very straightforward. You can see a leaf is green, right? You see your plant is green, right? Green comes from what? The chlorophyll, which comes from the chloroplast. Okay, so that's how you remember living cells, okay, for plant and animal. What will you see under a light microscope? Now, everything else you cannot see in a light microscope, you can see in the electron microscope. So for the pure students, it will be all of that. For the combined students, endoplasmic reticulum and Golgi is not in your syllabus. You will see mitochondria and ribosome, okay, and central lysosome. Right, so don't worry too much about it. Hey, can you hear my son? Clear? Now, this one over here, generally, they will test you more doing practical, okay? But in WA, if they want to test you, right, some of the parts, all right, what they like to test you on mainly is, what is this thing here called? It's the lens, okay? Then, where do I magnify and change things? That would be my objective lens. How do I adjust the thing? It will be the adjustment knob over here. So I have the big one called the cos, the very small one called the fine adjustment knob. Okay, but all intents purpose, if your school has not let you touch a microscope, or if your school hasn't really taught this a lot in your tutorial work, that's a hint. Probably not going to come up for WA. Okay? Now, this part I'm going to skip staining. You want to be tested WA1. Now, the different parts, okay, so I mentioned to you before, I'm going to teach you how to remember this whole thing like a Taylor Swift concert. Okay, and here's how I'm going to do it. To those who just joined us, hi, welcome. You can download the learning materials from the Zoom chat. So in the Zoom chat, there is a link. Please click on it and you will download it. All right, now back to Taylor Swift. So this one, now Taylor Swift, okay, when she creates all her songs, what happens? If I want to have a concert, who's the most important person? Taylor Swift. So from that one Taylor Swift, everything is going to project outwards. Okay? So Taylor Swift is the most important person to make the concert. She makes all the decisions. Likewise, in the cell, okay, what is the most important structure that makes all the decisions? It's going to be your nucleus. Then, like Taylor Swift, what is so important about Taylor Swift such that she can make music? All the music is found in her head. So in Taylor Swift's head, there is information about music. Same thing for the nucleus. It contains genetic information called DNA. Okay, it contains genetic information called DNA, which is where we have this point here, genes to carry out instruction for protein synthesis. All right. Now, the next part here about 23 chromosomes, father and mother. This one you already know from Sec 3, so I won't spend too much time on that. Now, back to the Taylor Swift example. When she wants to go through a concert, she will give instructions. Hey, someone book this particular concert hall. Hey, we are going to go to Singapore and not Thailand. Hey, this, hey, that. So Taylor Swift is the one giving all the instructions. Same thing like your nucleus. It will control all the activities of the cell. And that's how you remember. Nucleus controls cell activities. Now, Taylor Swift will sing and dance, but your cell cannot do that. The only thing your cell can do is just grow. So growth and repair. Okay. Now, this part here about condensing at this stage here is FYI for your info. The main focus I need you to know after today is the nucleus has genetic information, DNA, controls activities like growth and repair. And what is this information? It is the genes. Okay, so check the Taylor Swift. She is the middle of everything. She is the nucleus of her whole concert. Right? I will come to cytoplasm and cell surface membrane later. Next, okay, if I look at my structure of my cell, okay, I have my nucleus. Then what's touching the nucleus? Is these two structures, the RER and the smooth ER. So I'm going to go to those things next. So in my ER, okay, just like the Taylor Swift concert, what's going to happen? When she wants to create a concert, she must have two groups of people to help plan. One, the operation team. Why? You must go and set up for me the concert venue. So you set up the concert venue, put all the lights, everything. Wow, done very good. Then what is the other category of people who she needs help for a concert? It is her marketing team. Why? No one's going to know to attend the concert if we don't know the dates, the prices, and the venue. So Taylor Swift will have two people to help her. her marketing team and the ops team. And that is the same thing as yours, endoplasmic reticulum. As you can see in the picture here, the ER is beside the nucleus. So they are very important to the function of the nucleus. Same thing. Taylor Swift's marketing team and ops team are very important for a concert. They are directly connected to her, very close. All right. So now, okay, Cher, I see a bit of the link, but trying too hard, no. Let me convince you with the next part. So my ER, your will struggle. Wow, Cher got two, very hard to remember. How to remember two? So let me just guide you based on the end product. Okay. Now, my ER as well, my smooth and rough, end of the day, notice, oh, one settles protein, one settles lipids, which is fats and carbohydrates. So, end of the day, your function, you just have to know which one handles which. Okay? And if you go back to the characteristics, all right, now, RER is going to be like my marketing team. Okay, like my marketing team. Oh my God, the handwriting. Okay, I know some people here laughing, especially Zena. Okay. Now, why? When Taylor Swift wants to have a concert in America, the marketing team will make all the slides, all the poster, everything for the American audience. If I want to have a concert in a more conservative country, can they use the same advertisement? No, right? Cannot, ma. Okay, so that will have to change. So in order to change the material of the marketing, I must send my marketing material to somewhere to be edited. So that is why over here, okay, the RER, their job is to package something to be edited. What is the thing that will be edited? It is your proteins. Okay, so your RER, all right, is going to make the proteins. Okay, and where do I find the RER? Is continuous in the nuclear membrane and shape-wise, okay, it appears rough like pimples on my face because it has ribosomes there. So in the Taylor Swift marketing team, she has marketing people. Hey, let me turn the advertisement. Let me turn the advertisement. Making out advertisements for, okay, sending out to be modified. So that is the RER. Now the RER, ribosome, Mr. Paul, what is that? I just need to know in your head, okay? Ribosome, only got one function, site of protein synthesis. Over here, two subunits add together, don't need. This one, key thing is this. What it does is protein synthesis. Where I find it, two places. One on the RER, one floating around. Why? It's a bit hard for you to understand now, but basically, a cell needs proteins to be secreted outside. A cell also needs proteins for inside. Just like Taylor Swift, she must do marketing within America, and she must do marketing out of America. So that's why you have ribosomes in the cell and on the RER. Okay? Now, so that is the next part. So my RER will do all the marketing that needs to be edited. My smooth ER, this one is an ops team. Ops team just go there and make the theater, make the performance ground, job is done, they move on. So very simple, straightforward. That's why there's nothing extra on it. They just go there and set up. So for function-wise, okay, use the 3 biomolecules to help you. If I have 3, protein already synthesized by RER, what is left? The other 2 lor. And that is the hack for bio. A lot of things revolve around the 3 biomolecules, fats, carbohydrates, and protein. So one easy way to remember is, I remember protein for 1, the other 2 must be the smooth ER lor. Okay, it must be the smooth ER. Right? Okay. Then they detoxify the cell content. Okay, detoxify means what? Must clean up the rubbish. After every concert that Taylor Swift has, okay, the fans will go home, rubbish will be there. So the ops team who sets up the concert venue also has to clear up the rubbish. That's why the smooth ER is the ops team. Clear so far? Okay, then now, okay, at all concerts, okay, back to the part of marketing. So after I market my material, I need to change the advertisement, right? Okay, for my RER, what happens to my advertisement, which is the protein over here? It will be sent to the next organelle, which you all know lah, is the Golgi body. So just as I say, have to modify, okay, modify the marketing material. The Golgi's only job is to do what? Modify the protein. Hey, this guy's story starting to make sense. Of course it is. So the Golgi's job is to modify the protein for my RER. Just like the Taylor Swift concert. Her marketing team need to make advertisements and it must go to the next part, the Golgi to be modified to be suitable for different countries. Okay? Now, Goji, one very painful thing. Can you please put a star at the notes over here? This will sadly determine whether you get A, B, or C, or whatever grade you want, okay, in some cases. Why? Teachers like to test Goji. Okay, so if I were you, I will pay careful attention to the next 2 minutes I'm going to say. Check for a question. If you're being asked to label a Golgi apparatus, make sure it's a capital G. Not capital letter G, your teacher might just give you 0. It is G because it is some person's name, some famous dead person. So put a capital G there. Teachers like to test for WA1 Golgi body or Golgi apparatus. Clear? Okay. Now, then some of you, Mr. Paul, I don't know how to describe the structure of the Golgi. I don't know how to describe the structure of my RER or my ER. This one, very easy one. You can see the picture here, right? It is a lot of what? Sex. So that's all it is. It is just a sac that is made of a membrane. They are simply membranous sacs. The difference between the ER and the Golgi is this. You can see from the picture, okay, next one over here. ER, they are all connected. So if they're all connected, it's just like me on IG. Wow, I got a lot of friends. My network of friends are very good because I can connect to a lot of people. So connection means network. That's why over here, hey, it's a network of membranous sacs because they are connected. Then Golgi, how come I don't see the word network? I see flattened. Because as you can see in the picture, this is like your Wi-Fi symbol. The lines are not connected. So this one got no network, that one got network. That's all. Easy peasy, lemon squeezy. Okay. Now, how the different parts work together, this one sometimes will come out as a structured question. Okay. I want to guide you here. It's very, very straightforward. Go with the marketing story that I said. It starts with the RER. So I have the protein, my RER. The RER protein, okay, will butt off from my RER, go to my Golgi to be modified. Basically, my story. The advertisement must go to somewhere else, the advertising company, to be modified. After the advertisement has been modified, what does Taylor Swift company now do? Wow, I advertise to the world. Boom, it advertises to the world. That's why in Singapore, we have that concept poster of her in the very sexy dress at the MRT station. There are a lot of boomers go, not very demure. Okay, so same thing. After the protein has been synthesized and modified at the Golgi, it will be secreted out. That's all. Okay, so start synthesis at the RER because ribosome is there. Goes to the Golgi to be modified. Okay, all right. Then after that, it is released. Now, Akshaya asked a good question. Is there a specific site that we need to memorize, like which part is being added to? Now, strictly speaking, in the O levels, there isn't. But if your school teaches you the site, which is the cis and the trans, then your school is over teaching you. But they'll probably be testing you for your exam. Okay, so at O levels, you don't really need to know the names of the site. But if your school teaches you, it is going to be the cis and the trans. Now, how to remember which one is which? Okay, alphabetical order. C to T. C comes first, so enter cis phase, leads through the trans phase. Trans phase for transport out. That's how I remember it. Okay, so I hope that helps you. Okay, but that's a brilliant question. For the rest of the words, what did this guy just say? Oh, it's not relevant to you, so don't worry. Okay, now back to my Taylor Swift concert. At the concert can only happen if got Taylor Swift, if got the fans, and must have what? Power. Can you imagine a concert? She's up there on the stage, everyone is screaming, sing all the song, but then there's no lights, there's no camera, there is no music and no microphone. GG. Need to refund money. So where is power going to come from? The mitochondria. This one, enough said. I don't have to talk so much because all of you grew up in the meme era. I just say this now. I know you will say it in your head. Repeat, complete the sentence. Mitochondria is the... In your head, you just went, powerhouse of the cell. How come I know that? Don't worry, you know that because it's a meme. So mitochondria is the powerhouse of the cell. Just like in my Taylor Swift concert, they need energy for all the lights, all the generator. So that is going to come from my mitochondria. Okay? Then the shape, this one is like your jelly bean. Okay? It is more similar to the pineapple tart in Chinese New Year that is long, round. That is called rod shape. Rod shape. Alright? Now, in a different color pen, put a double star here. Mitochondria has a double membrane. Okay? Then, next, put a star at the end here. So just like I told you, right, the teachers like to test about Golgi body or Golgi apparatus because of the capital G. The second organelle that teachers like to test students is the mitochondrion. Why? If the question paper point only to one, oh my goodness, here don't have, okay? If they point only to one, they expect in your answer to be mitochondrion. If they point to two, you must write mitochondria. So this N and that G in Golgi, sadly, my experience is the number one way how people lose a mark, and always very sad by karma, okay? They lose their mark, right? Then they get 59, 64, 55, no, 54, 49. That one mark would have gotten them to a new grade. So please pay attention to this. Okay? Now, mitochondria, straightforward. Powerhouse of the cell. That is the story. In science now at Sec 3, you have to say, what is the process that gives me energy? Respiration. Okay? Respiration. If your school has taught you aerobic respiration, go ahead and say it. Usually, they only need it as an answer after they teach you the respiration topic. Because in your head right now, you have no idea what's the other respiration. Okay? So what's respiration? You eat all your sugar, pineapple tart in Chinese New Year, digested, broken down to give me glucose. Glucose is broken down to release energy. So this one put another star here, okay? The physics people will kick you if you say produce energy. Bio people actually don't care. Okay, to us is, yeah, release, produce, nothing wrong ma. But the physics people like, cannot, energy can never be produced, only from the sun. So we, okay lor, we release energy. So your school teachers will probably also mark you down if you put the word produce. Okay, because at O levels, they will mark you down. So take note of that. Okay, so far there are 4 stars. Okay, now the mitochondria is the one of the organs with a double membrane. The other one, okay, is going to be your nucleus over here, right? But double membranes are not so important in your syllabus. Okay, now TLC done, ribosome done. Okay, now we go back to some of the earlier organelles over here. Okay, so inside the telecive concert, okay, if telecive is the nucleus, and I'm doing advertisements within my country, the cell will be America lah. Okay, the country now that's not so great because of Trump. But long story short, America has a border to protect itself. So what is the border of the cell? That is the cell surface membrane. All right, and you have to say the word selectively or partially permeable. Why? Look at Trump right now. To him is, some people can enter. If I'm a Singaporean tourist with tons of money and I go there and spend a lot of money at that casino, please come in, we want you. But sadly, if you are a displaced immigrant from a country that is war-torn, sorry, please don't come. We don't welcome you. So there is some selectivity there. It's partially allowing some people in and not others. Hence, use selectively or partially permeable. Why is semi wrong? Semi is like, okay lah, I let half the Singaporean in, I let half the refugee in. Half. No way. Mr. Pua cannot half go in. I will die. Okay. And just as how it controls movement into America, the cell surface membrane controls movement of substances into the cell. Okay. Then within America itself, where Taylor Swift is driving around having all her concerts, okay, that is the land. Same thing. Cytoplasm is the top part of the cell. What happens to be in the cytoplasm? Anything that is soluble in water that you have learned from Sec 2 digestion. Things we take into the cell that are liquid soluble are found in your cytoplasm. So that's why it's mostly water. You are mostly water. And yes, things like carbohydrates, lipids, and proteins, as well as all your different organelles that you have just learned. Okay. Now, if they ask you cellular reaction that occurs here, just say respiration. Okay, you can just say respiration. Okay, no, never mind. Sorry, don't confuse you. Respiration in the mitochondria. This one, you can just say glycolysis. But this time they won't retest you. It's 2 out of your syllabus. All right, so now that is the telecive example finished already for animal cells. Okay, plant cells is just adding on a bit to that telecive concept lah. Okay, now if telecive is doing a concert in a country that has more security means what? Their borders are more secure. That is like the cell wall. So the cell wall is only found in plants. Okay, it's fully permeable. Okay, idea is you must remember that it's fully permeable and it's different from the cell membrane because there are cellulose fibers. So when you're answering anything for plant cell wall, put in cellulose fibers. Okay, and as the name suggests, it's a wall. So wall means what? Any wall is meant to protect you and separate you from something. So protect the cell, prevents the cell from bursting. which we will touch on later in the next part of the topic. Okay. Now, if you don't know, okay, this cell wall is the reason why when you bite vegetable, it is crunchy. That okay, or yesterday or 2 days ago or 3 days ago, you eat low hay, right? You bite the carrot. Why so crunchy? You are literally biting cell wall. Okay. Then cell wall allows anyone in. Okay, totally anything can enter the cell. Only your cell membrane is partially permeable. Alright. Then in plant cell, got chloroplast lah. This one very straightforward. Okay. This one is like a Taylor Swift concert. Okay. She want to go green. She want to help the world. I don't want to just keep burning fossil fuels. So what she do? I put solar panel. I put solar panel, right? Daytime, the light shine, booms, I suck and absorb energy so that my carbon footprint is positive, so that all my fans will love me even more. So that's what the chloroplast is like, a solar panel. It will absorb light energy and convert it into something useful. Light energy to chemical potential energy. Booms. Sec 2 math, Sec 2 science here to haunt you again. So what is photosynthesis? Is the reverse of your respiration. I want to produce glucose using carbon dioxide and water. Okay, then they give us oxygen so that we can live. All right. Now, notice over here the chloroplast, the shape sounds very similar to my mitochondria. One is oval shape, one is rod shape. Pretty similar, long one. Put a star here, you then realize, oh, both are double membraned. So what's the trick here? Any organelle in the cell that has to do with releasing energy, okay, or creating glucose, which is energy, both are double membrane. Okay, that's the trick. Anything to do with energy, that organelle has a double membrane. All right. Then what's the function for? For photosynthesis. Okay. Now, next one, vacuole. This one, okay, is literally, all right, just a water ball. It's a water ball. So in animal and plant cells, a bit different. Animals need a lot of water. So animals just have one large permanent vacuole in the middle. Okay, it's like their reservoir. Animal cell got many, many 小球球, okay, many small vesicles around. That's all. Okay, that's all. Sounds straightforward. Over here, very cheap, don't worry, you will cover more in the transport of substances. So I won't go too much. But vesicle, just take note, single membrane, plants got one big one, animal got many. Both do the same thing, store stuff. It just store stuff. Just change the word stuff with glucose, mineral salts, or whatever the question is asking you about. Okay, ribosome settle. And yes, we are all done. All right, so here we have some analogy. I never use it because between Taylor Swift and the chef story, Taylor Swift every single day. You might be thinking, Mr. Paul, are you a Swiftie? No, whenever I hear Taylor Swift songs, I swiftly run away. Haha, get the joke because I'm a dad. So it's a dad joke. I don't like her songs. I like two only. I find her jokes a bit too repetitive. Okay, now level of organization, this one is literally you once again. So if you have paid attention in Sec 2, this is a bit easier. But let me just do a very quick recap. Right? Now, first thing first, cell is not the smallest, it is the organelle. So I have my organelle, then I have my cells. This one, usually they test you in MCQ. So very, very hard, okay? Don't come out structured. I haven't really seen it come out as a structured question. They usually test in MCQ, what is the correct order? So I just need you to know this. The difficult one that students have is tissue and organ. Okay, what's the difference? Now, tissue is like your tissue paper. Students are confused. Tissue got different kinds of tissue, different kinds of cell together, or only one type of cell. Now look at your tissue paper. When you blow, right, that tissue paper is made of the same type of plant fiber. Either all white or brown, okay? Don't have halfway got gold threads or metal threads inside. It's all just paper. So same thing, what is tissue? Tissue generally, right, okay, is, okay, similar cells performing similar function. Okay, now complex is only mostly for blood lah. So that's an outlier. But in your MCQ, right, generally just know cells is the original number one. I put many cells together, I get simple tissue, like muscle tissue, okay, skin tissue. Then, okay, stomach, pancreas, liver, that is the organs. All your organs are many different types of tissue working together. Why? I have my stomach cell. I have my nerve cell. I have my blood cells. Okay? Because I have different tissues working together. Organ is the one where it gets confusing. So all you have to know is anything if organ is like rojak. Different. Before organ, Okay, at tissue, everything is 1, 1, 1. Cell is just one cell. Tissue is mostly only similar one type of cell. Organ and organ system is just different things before that. So organ is different tissues, organ system is different organs, organism is different organ systems. And that's how you make it more and more complicated. All right, but these are mostly in MCQ. So don't worry too much, mostly in MCQ. Okay. Now the specialized cell one, this whole entire thing can be summarized as one thing. Okay. Everything in the animal cell and the plant cell, whatever structure there is, if in exam you blank out, just remember, hantam this word, increase surface area to volume ratio for rapid whatever. And I'm going to prove it to you. Red blood cell, biconcave, for what? Increase surface area to volume ratio. Okay. Root hair cell, got extra long protrusion, for what? Increase surface area to volume ratio. So if in the exam, they ask you for a structural feature for either of it, whack the structure that gives you an increase in surface area to volume ratio. Okay, high chance you are correct. All right. Then now red blood cell, this one is kind of like this. You go to school. If I go to shopping mall now, right? I go to NTUC. They say, hey, you have just won the lucky draw. In 1 minute, whatever you can fit into your bag, you can bring home. Would you bring a bag that has all your homework, calculator, school uniform, track shoe, and iPad? Or will you bring an empty bag? You bring an empty bag, right? To maximize. Same thing. That's why the red blood cell got nothing, got no organ, got no nucleus and other organelles. Why? So it's empty and it can pack more hemoglobin into it to transport more oxygen. Okay. So hemoglobin is the protein in the red blood cell. All right. Okay, how to remember the lack of mitochondria? This one, classic example. How many of you order Grab food? Then when the Grab food reach your house, how come my pizza, large pizza, got one slice missing? What happened? The Grab driver eat my food. Wow, like that, not good lah. That's why I can't wait for the day AI happens. Then the AI deliver bot will deliver the pizza to my house. The AI bot won't eat my pizza, ma. But the red blood cell is big brain, is higher order than us. It has already solved the solution. My mitochondria will use the oxygen. If you use the oxygen, then I cannot transport the oxygen. So what I do, I just remove my mitochondria. That's how you remember it. My red blood cell needs to transport oxygen. So I just remove anything that will use my oxygen. Okay? Then at stage you're wondering, sure, then how does it undergo respiration? Don't think so much. Okay, this one will cover at A levels. For WA1, we survive first. Then we go back to that. Okay? Intestinal cell, same thing. Got a lot of microvilli. Okay? To do what? Increase surface area to volume ratio. So like I said, in the exam, write this down if you don't know. Then the next part, increase surface area to volume ratio for efficient what? Absorption of nutrients, diffusion of oxygen for the red blood cell, root hair cell, okay, absorption of water and mineral salts. That is called contextualizing. Okay? Right. Then for xylem, this one, you all drink bubble tea before. Right down here, xylem vessel is a bubble tea straw. It is a bubble tea straw. So bubble tea straw, what are the good parts of a bubble tea straw? My bubble tea straw is long and hollow so that all the bubble tea pearl can reach my mouth very quickly. Okay? Same thing here, long and hollow so that there's a continuous smooth passage of water from the roots to the plant. Then your bubble tea pearl, I know there are two kinds of straws. Sometimes they give you the lao ya straw, right? Then you poke, poke, poke. What happened? The straw keep bending. Wow, I hate that kind of straw. But if they give you the very good straw, poke one time, whew, very, very good. So your xylem is the better straw, okay? So it's very strong because it doesn't want to collapse. And what keeps it strong? This lignin deposit. Okay, so this lignin deposit helps to prevent it from collapsing. All right. Okay. Okay, I hope you are following. Okay. All right. Hi, Dina. Welcome. The links for the materials today are found in my Zoom chat, so please download it. All right. And with that, we are going to go to the questions over here. So what I'll do is I will go through it. Okay. What I need to learn from me is this. Look how I break down the question. The difference between an A grade student or B grade okay, versus student who doesn't do as well is they rush into the question. MCQ, I want to change your mindset. It's not about you finding the one correct answer. Sometimes very hard to find because you never really study. But you realize, if I ask you this, MCQ is finding the 3 wrong answer. Wow, life becomes very easy. You will see life becomes very easy. So you choose the answer that you confirm know is not wrong. Okay, so cancel out those that are wrong first. Let's start with the first one. Now, the diagram represents the cell structure of the human liver cell, okay, a leaf palisade cell, and the human red blood cell. Which cell structure is X? So, straight away, first thing first, okay, I got human liver cell, human red blood cell. Which structure here can I delete? Okay, my human cells can have cell wall or not? No, booms, out. Can have chloroplast or not? Booms, cannot. Then, nucleus. This one got nucleus, red blood cell? No nucleus, Human liver cell got nucleus. Leaf cell got nucleus. So sure I know, nucleus is not there. Answer is cytoplasm. Oh. So you see, I cancel out what I know is confirmed wrong first. Easier. Oh my god, wait, I sent to the wrong person. Okay, people who just joined, sorry. Please download the link. I'll send the link. Okay, sorry, just now I sent the link to a personal message. Now, yeah, so the links for the materials I'm going through now can be found in my chat. Do download it in the Zoom chat. So go to the Zoom chat and download the link. Next, point number 2. Okay, this one of the toughest questions. This topic is the toughest question when it comes to this cell structure. Why? Wow, the kids said, radioactive amino acid. Why is this very cheap? This one, very simple. When you see radioactive, just imagine glow in the dark. Oh, so yes, the amino acids are glow in the dark, which means what? If I put them in the cell, it's very dark, I can't see anything. Hey, I can see where they're moving around. So I can see where my amino acids are moving around, that's all. It's like a glow in the dark, or it's like air tag. Okay, so this question is just asking you, if my amino acids were to move, where would they appear? So the question will be, where they appear last, where they appear first, where they don't appear. Let's look over here. Where will it appear first? Okay, so this is linked to protein synthesis. Then I need to recall, what are the structures that will synthesize proteins? It is my ribosome. Then, which other organelle has my ribosome? My RER. All right, but this is a tricky question. Okay, where will it first appear in? Okay, where will it first appear in? Now, after the protein goes to my RER, it's like my Taylor Swift thing. I synthesize in my RER, okay, for marketing. Then marketing, before it can go to Singapore, it must get modified. So it gets modified by the Golgi body. So this is my order from ribosomes, RER, Golgi body. Now, will there be, okay, any involvement of the nucleus? No. No protein synthesis nucleus. Nucleus, okay, it's like me asking you, you think Taylor Swift will again take part in creating the marketing advertisement, man? No, Taylor Swift so rich, she can sing song. She will not touch the marketing material at all. She pay money, someone else do it. So Taylor Swift only sing song. Nucleus only control the activity. So no protein synthesis. So gone. Okay. Cell membrane. Okay. This one here is saying, in my thin cell membrane, will the amino acid get stuck there? Okay. It won't lah. It just goes through it. So it cannot be my cell membrane. Then it's between RER and Golgi. Then remember the story. It must first be created, then get modified. So RER first, Golgi is wrong. Answer here is 2. Okay. Now I give you a pro tip. If your school got this option, which they love to put as a trick question, they put cytoplasm. Okay. Option 5. If got option cytoplasm, then I ask you now, is it cytoplasm or RER? Which one is the answer? Okay, the answer is the cytoplasm. Why? Here's the thing. If I'm printing posters in America, I need paper. Correct? So what happens first? The paper must be imported. When it's imported, it's just on the roads. Same thing. When the radioactive amino acid enters the cell for the first time, it is floating in my cytoplasm. So it enters my cytoplasm first, float. Okay? So I go to the picture, it will float all the way here, float, then enter my RER for usage. So this is how the school can trick you. Okay, so let's be very clear. Is there cytoplasm there or not? Okay, check for cytoplasm. Now, question 3 over here, same thing. All right, which structures can be seen in a human sperm cell when viewed under a light microscope? What's the trick? Light microscope, I draw what I can draw in a cell. What's my art skills very lousy? I got the cell surface membrane, I got the nucleus, sperm cell, I just draw one tail lah. So that's all I can see. Human sperm got chloroplast not? Please don't tell me have. We are not plants. Okay, we don't shoot seeds. Okay, we shoot sperm. Okay, rather male to produce sperm. Then ejaculated is sperm. So there's no chloroplast inside here. So once I know got no chloroplast, life is easy. Then now look at this. What can I see under a light microscope? I already know I can see my nucleus. So if I know I can see my nucleus, this one straight away is gone. Okay, then I only can see my nucleus. I cannot see these two. So life is easy. If I cannot see these two, I will see my answer here is D. Okay, answer here is D. All right. Question 4. Which organelle is not correctly paired with its function? So this one, okay, once again, you must know your function, and I hope my story will help you. Okay, I hope my story will help you. So let's go from the bottom. Ribosome joins amino acids together. If I join amino acids together, what do I get? I get protein. Eh? Just like how I take Lego bricks together, I'm building up a Lego figurine. If I join amino acids together, I'm building up a protein. Hey, this one makes sense lah. Alright. Mitochondrion, easy peasy lah. Releases energy, confirm correct. Golgi apparatus or Golgi body. Okay, the last part of the marketing material. They will release the vesicles that contain the proteins. It will release the advertisement. So releases membrane-bound vesicles. Yeah, that's correct. And endoplasmic reticulum. Store carbohydrate. Nah, okay. Doesn't store carbohydrate. All right. It can help synthesize the carbohydrate if it's a smooth ER. So it doesn't store carbohydrate. Here is wrong. So answer here is 1. A. Clear? Okay. Right. Okay. So in lieu of time, I'll be skipping. Yeah. Okay. I go to this structured question over here. Now, this part here, I want to teach you something very important. Okay. When I see a structure like this, how to identify the mitochondria? Look at this rod-shaped cell. Okay, it will look like a jelly bean. Next, inside got lines going this way. When there are lines going this way, or you see shape over here, this is the mitochondria. If it's a chloroplast, it will be like that, and the lines will go lengthwise. So that is my pro tip to you. Okay? If it's lengthwise like this is chloroplast. If it's horizontal, okay, like this, or it forms a ellipse, okay, that is your mitochondria. First tip. Second tip is this. For every question on cell structure, the school paper will tell you this thing. Animal or plant cell. Make sure you double check. Okay, if they don't tell you this, they will give you a very clear cell wall. Then if not for that, you won't know. Okay, then identifying wise, okay, if I all the time the world, I'll ask you more stuff, but a bit tough, I cannot. So here, nucleus, okay. Over here, A is my cell surface membrane, B is my RER. How do I know? Because on the surface of it, they zoom for you, that is the dot on top. So that must be my ribosome. Now let's be clear. If they zoom in and point to the dot over there, the answer is ribosome. But if they point to like this, C, what is this? That is my RER, or rough endoplasmic reticulum. Okay, so that is the difference in answering. If they purposely zoom into the dot, it is the ribosome. If they never zoom in, it's the whole thing, it is your rough ER. In case you don't realize, you cannot write RER. I say because no time, but please spell out rough endoplasmic reticulum. Okay, same thing for Golgi apparatus. Okay, so A is your cell surface membrane. What's the function? Okay, to prevent and control things entering the cell. I won't spend too much time on that. Now, B is my RER. What's the function? For protein synthesis. So don't worry, okay, the answers we will flash later lah. Okay, you can take a picture of it. Now, part C. Liver cells have more mitochondria than most cells in the human body. Suggest why. So what's a suggest question? Suggest question means you never got taught it. It's not in my notes. But you kinda are able to apply the answer based on what you have learned. So when you come to suggest questions, I know kids are very scared. Don't be afraid. First thing, just tell yourself, I confirm learn before one. You just need to find out what you learned before. So look at this, analyze. Mitochondria. What does mitochondria do? Oh, it releases energy. Okay, so I know if I have mitochondria, I can release energy. So if I have more liver cells, obviously I can release more energy. So the question here is, why liver cells have a lot of it? You just have to think, what is one function of the liver? Okay, so if you remember from digestion in Sec 2, the liver is involved in what? Oh, producing, okay, bile. So you can write there, liver has to produce bile, that takes up energy, and therefore it needs a lot of mitochondria. Later, when you learn digestion in humans, this part, you will learn all the different processes of what the liver do. Detoxification, okay, deamination. The real answer here is the liver does one of that processes. Hence, it requires a large number of mitochondria to release energy. That's all. So I'm teaching you here how to contextualize the question and break it down. Okay. Now 5D, this one very easy peasy lah. State two structures. So command word state means your answer is very short. Please don't write one long answer like explain or describe. State is very short. So two structures which are found in the plant cells but not shown here. Your answer can be as short as just one word. Okay, so which two structures are found in a plant cell but not here? Straight away, easiest low-hanging fruit. Cell wall. Okay, chloroplast. Alright, so this is testing to compare between animal cells and plant cells. But once again, the key learning point for this question is state means you don't need to write a very long answer. I can just write two short answers like this. Alright. Now, then in your WA, probably there'll be some question, confirm have one question about absorption one. Either in root hair cell, okay, your red blood cell, or in your epithelial cell. So I just need you to look at the picture and realize, regardless of whatever it is, the cell there will increase surface area to volume ratio. Okay, whichever the thing is. Here, there's microvilli. So on the surface over here, once I see microvilli, hey, increase my surface area to volume ratio. Then suggest the purpose of it. Go back to this. What is the original function of the small intestines? Original function is what? Absorption of digested nutrients. So for 2 marks, what's the two key idea? Oh, I have a lot of microvilli to do what? First idea, increase the surface area to volume ratio. Second mark, so that there is more efficient absorption of digested nutrients. And that is all. So easy? Yes. Once you apply proper question analysis, exam questions become very easy. You become excited to do it. Okay, that's a bit overmuch, but yes. Okay, so I hope you can, for me today modeling, you can start to do a bit of question analysis to break the question down. All right. Okay. Then now moving on to move more substances. This topic sounds very long. Okay, but I'll have you summarize into 3 different things, okay, that overlap very closely in a short while. So I don't need to remember so much. I just need you to know, okay, 3 different things. Okay, 2 different things. First thing first, in this question, or these questions for this topic, step number 1, write this down. Step 1, identify osmosis, diffusion, active transport. So before you start the entire question, right, look at it, read it. Is it diffusion, osmosis, or active transport? Once you identify it, it's just plugging the correct English terms to put in the answer. Okay? Step 2. Okay? Identify the different regions, higher and lower. So you identify in the question, which one is the higher water potential, if it's an osmosis question. Which one is the region that's a lower water potential? If it's a diffusion question, same thing. Which part is higher concentration? Which part lower concentration? Same for active transport. So you label all of these things first. Okay. Step 3. Okay. Draw an arrow on the diagram to indicate direction. So this is the most common reason why students lose marks for this topic. Okay. It's not because they don't know. It's because they see the question, they do all the thinking in their head. Then when they write, they forget what they think. So later I'll show you, just draw the arrow. Is the thing moving in or the thing moving out? Then as you describe your answer, I just follow the arrow. Very easy already. Okay, once I draw the arrow, go in or go out, your answer becomes very easy already. Okay? So this is generally the 3-step process to doing well for this whole entire topic. Identify first which process is it. Identify my different regions, which one higher and lower concentration of water potential. Then third, draw the arrow to show the movement. You do all of this, you write your answer, confirm correct one. Okay? Now, over here, I will summarize diffusion for you in a very simple nutshell. Okay? You cannot smell, but now if I fart, what happens? After a while, if you are in a classroom with me, all of you will smell it. Because why? Your fart diffuse across. So when I farted right outside my pigu, what happens? There's a very high concentration of fart molecules, which is my what? Step 2. Identify the different regions. Then, where you are seated, the area near your nose has a very low concentration, in fact, zero, okay, concentration of my fart molecules. Then you know lah, diffusion is along a concentration gradient. So what's going to happen? You just write your answer. The fat molecules will diffuse from a higher concentration to a lower concentration. So region of higher concentration to region of lower concentration along a concentration gradient or down a concentration gradient. All right. Now, after I fart and the thing goes to your nose, okay, what happens? Do I not smell fart anymore? No. Halfway you will say, oh my God, Mr. Pua, you fang pee, very smelly. I say, ha ha ha, yes, I did, sorry. So when you're suffering, I'm also suffering because what? I can still smell because not all the fart molecules have gone there. So that's the idea of net movement. Some go there, some come back. Okay, so that's why the word here is net movement of particles from a higher region of concentration to a lower region of concentration down a concentration gradient. So a good quality answer for this topic will have 3 key parts to it. Okay, the net movement, number 1. Then the particles from high to low or low to high for transport, number 2. Then third one, down a concentration gradient or against a concentration gradient for transport. Okay. Then next, what factors affect the rate of diffusion? This one, very easy. Back to my third question. If got a fan down there, you'll blow faster, right? Then the thing will spread faster. So, same thing. If there is something to increase the speed of movement of molecules, what will increase the speed or movement or energy of particles? Temperature. Okay? So higher temperature. Right? Now, if I fart a loud fart, very long, at 5-second fart, wow, got so much particles there, there will be a larger concentration gradient. Lower at where you are, steeper concentration gradient. Okay? Then, large surface area to volume ratio, this one, you really know this from my absorption of my root hair cell, blood cell, okay, as well as my microvilli. If there's larger surface area to volume ratio, increase speed of diffusion. But don't need to remember so many. Increase, remember the temperature one, okay, and the concentration gradient one. 1 to 2 to write in your answer is more than enough. Now, some things will not be able to diffuse. I need you to remember this, okay? So I just draw and show you. Your glucose, of everything here, your glucose is the largest. So glucose, draw with me, it is a 6-sided thing, a hexagon. So this is the limit, okay, in your this topic. They will put some glucose or some sugar solution in a whisking tubing. Then the glucose can diffuse out. Sometimes they will trick you. They will say, oh, I put in starch, sucrose, and proteins. All of these are bigger than this. So how do I draw sucrose? Sucrose is 2. Okay, is 2 of my glucose joined together in a very rough approximation. So A2 is larger than this. What is starch? Starch is this thing times 1000. So confirm very big. What is proteins? Proteins is 1000 amino acids. So if my amino acid is the smallest that's accepted, 1000 of it, wow, very big, cannot what? So when it comes to those questions, case study, especially with the beaker and the whisking tubing, check. Did they put any of these 3 things? They are out to cheat you. Okay, they're out to cheat you. All right. Now, over here in our notes, you got some very nice answering technique teaching you how to answer. Okay. So like what I said, you know the regions already. State which one has a higher or lower concentration, how it moves, enters the cell along a concentration gradient across a partially permeable membrane. So this one you all can get and read. Okay. I won't spend too much time on it. Now, what I will help you with the next part. Okay. Now for water osmosis, it's the exact same thing. I just replace particles with water and molecule. So put a star here. How do students lose the marks for osmosis? Okay, it is they don't have the word net, they don't have the word molecules, and they forget to change it to water potential. Okay? So you must use the water potential. Now, osmosis compared to diffusion, what's the main difference? Okay, this one need a partially permeable membrane. Why? Now, diffusion don't need membrane. Just remember, I fart, you sit so far also can smell. There was no membrane there. Okay? But water, why I need a membrane? Because you see, if I have a beaker of water over here, okay, I got a beaker here. Then, okay, I have my whisking tubing. Then my whisking tubing inside here got stuff. Because there are two separate components, I can say that this one has a water potential number 1, here as water potential number 2. Then I have two different regions of water potential. One has lower lah. But if I remove my partially permeable membrane, what happens? Then my water molecules at 1 and 2 become the same mass of water molecule. So I don't have two different regions of water molecule now. I just have one large region. That is why you need a partially permeable membrane. And then oh, yeah. Okay, right? So the trick with questions on water potential and concentration is this. Sometimes they will give you a water potential question, tricking you, okay, when actually it's a concentration question. So what you do here is this. Once you find out there's distilled water, pure water, okay, water potential is very high. If I have any kind of concentration, percentage 1, 2, 3, right, this one means water potential is low. Water potential is low. So look at the question. Sometimes actually a diffusion question. They're telling you this thing is distilled water, that one is 20%, okay, salt solution. Remember yourself, okay? 20% water potential, 20% salt solution means it is low water potential. So that can be a diffusion question or an osmosis question. So take note, anything that has a percentage as a concentration, yes, it is concentrated. It also means it has a lower water potential than distilled water. Now, this next part over here is the one where you all really have difficulty remembering, okay? So let me help you with it. Isotonic, this one, your 100 plus, H2O, all of these are isotonic. When you drink after you exercise, why? So that my hydration level in my cell becomes the same as before. Okay, I don't want too much, I don't want too little, I want isotonic. So that's how you remember. Isotonic is just right. Same water potential. So when there's same water potential over here, there is no net movement of water molecules. Then, I can't remember which, either hypotonic or hypertonic, which one is the one. Okay, so here I'm going to help you. Okay, and I know a few people who have attended my class before, you know what's the method, and it's damn simple. You actually learn the answer to distinguishing hyper and hypo when you were in P1, when you learn the alphabets and you learn numbers. Because why? What is the difference between the spelling of hypertonic and hypotonic? All of you will know, or just the E and the O lor. Okay, so here's the first trick. You write down what is the number for E in the alphabet. A, B, C, D, E. So E is 5. Okay, then hypotonic, what is the number? O. Can count. A, B, C, D, E, F, da, 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 da. Okay, after you count it in your head, you realize O is number 14. So, 1 is 5, 1 is 14. So 5 and 14, which one is lower? Hyper, right? Lower. Okay. So if it's a hypertonic solution, it's lower, it's more concentrated. Okay, it's more concentrated. Because why? It has hypertonic means the water potential. So 5 is lower, it has a lower water potential. Then you don't believe, we check the other way. Okay. Now if I put it in a hypotonic solution, here is higher. Higher, right? Higher water potential. Oh, higher water potential in the solution, my cytoplasm has a lower water potential. Oh my God, it checks out. Of course it checks out. Okay, because I spent so much time making this, so it's easier for you. So just use the number. 14 is higher. So hypertonic, higher water potential. Hyper, E is number 5. 5 is lower, so lower water potential. So, okay. Then next one, you're confused is, animal cell, plant cell, which one do what? Okay, I know animal cell, more water enter. So you go animal cell first. When a lot of water enter the animal cell, what happen? It will burst or lyse. I like the word lyse. Lyse sounds more intelligent. Okay. Then when it enters the cell, okay, when it shrinks, it becomes the word crenate. So either you use shrivel or crenate or shrink. I like the word crenate. In fact, if I were you, I'll put crenate. So how to remember? Wow. Sure. animal cell create, sometimes I confuse with plants. How to remember? Very easy. Use the acronym ACL. So ACL is very relevant to you all because most of you generally, not judging, okay, you all do ACL every day. What is ACL? ACL stands for always chasing likes. Correct? CNY, you post your OOTD. Hey, go and like, go and like. Some people are chased for likes. So when you post stuff, okay, we are always chasing for likes. And that's how you remember. Animal cell, create likes. So in case it's not clear to you, animal cell, create lice. So my animal cell, I either use the word create or lice. Okay, lice means burst, create means shrink. Okay, then because you're Gen Z, that's easy for you. Now I come to, okay, plants. Sure, plants how? Okay. Now, plants, same thing. We always chasing likes, right? Where do you post to chase to get your likes? So for plants, what are the terms? Plants, okay? All right. Okay. So, okay, this one, yeah. So plants will be plasmolyzed when it leaves the cell, so it shrinks. So just remember, if the animal cell one is crenate, okay? Animal is crenate, it shrinks. The plant one is going to be plasmolyzed. So that's how you remember this, okay? One acronym to help you remember which one I use for animal, the other one, plasmolyzed, I leave for plants. Now, this next one to help you remember what is the shape of the plant cell. When my plant cell got a lot of water, it becomes very turgid. Okay, become very turgid. This one, it plasmolyzed, it becomes very flaccid. So PFT. Okay. So PFT, all right. PFT, all right. You can just remember poof, the sound, but that one a bit funny. So same thing, you're always chasing for likes by doing what? By posting, okay, for TikToks. You post for TikToks, okay, to chase for likes. So plants, turgid. The P here is turgid. Okay, here is flaccid. Okay, so when water leaves the plant cell, it can become plasmolyzed or flaccid. Then P reminds you plants. And that's how you remember. ACL, always chasing likes. Animals, crenate, lice. Plant cells, okay? PFT, posting for TikTok, okay? Plants, flaccid, turgid. Hypertonic, number 5, lower. Number 14, hypotonic, higher water potential. And if that, this topic really very straightforward. So many words here, but if you remember my 3-step process in front, plus all these little acronyms and stories, well, life will become very easy. Okay, all right. Now, what's active transport? Active transport is the reverse of my diffusion. Okay, now this one need to have a cell surface membrane. Okay, why? Like the osmosis, without the membrane, can I have a difference in concentration? Cannot. Okay, so for this topic, okay, when it comes to an MCQ, what is a very big hint that it is active transport? Okay, I will lead you to the idea that this requires energy. So what's the energy going to come from? Energy comes from respiration. So what do I need for respiration? I need oxygen. So if there's a question, they tell you, one got oxygen, the other one, the process no oxygen, straight in your head, this is an active transport question. Okay, take note. This is an active transport question. Or they tell you, this thing got a lot of mitochondria. That one don't have. How come the one mitochondria can do this process? The other one cannot. Booms. Mitochondria is to release energy. Once again, respiration. Once again, active transport. So that is the tip to answering this part about this topic. Okay, so let's go to the question here. All right. So first question, if you pay attention to me, when you read the first line, what is the first thing that will give you alarm bells? Down, down, down. Hey, the beaker shows it contains sucrose solution. Glucose is 1. Sucrose is 2. This is too big. So the sucrose is here. Okay. This bag contains sucrose. Can sucrose go out? No. So step 1. Should I tell you a question is this? You ask me to conclude. Okay. What are the sucrose concentrations over here? Step 1. Is this a diffusion question or osmosis question? Sucrose cannot even go out. So if sucrose cannot go out, it's not diffusion. This must be a osmosis question. So it's osmosis question, right? Now, what's happening? What is the higher region? Which one has more water? Which one has a higher water potential? Okay. The sucrose solution, yeah. Okay. The one in X or the one in Y? We don't know. So you must look at the question here. At the start, okay, of investigation, the bag was weighed. Then after 12 hours, it weighed 8 grams less. So if I have two regions where there's sucrose solution and my sucrose cannot enter or cannot leave, what is moving? Water. So now you here, after 12 hours, it weighed 8 grams less. If it weighs lesser, third step, draw the arrow. Oh, it means water has moved out. Water has moved out. So if water has moved out, I can label my regions now. So water, okay, molecules in osmosis will always move from higher to lower. So now I'll label which one is higher. Oh, Y is higher. Okay, sorry, higher water potential. Here is lower water potential. So now I know higher, I know lower, I can go to the question. Okay, so at the start, okay, X is higher or lower than solution Y. Okay, now which one can I delete? If my X had a higher water potential than solution Y. Okay, now they know that's water potential. Now I'm looking at concentration of the solution. So wow, tricky. So if sucrose has a higher water potential, what is the concentration? This is what I mean just now. You must be able to reverse. So higher water potential, low concentration. So Y has low concentration. X has the reverse. X has high concentration. So I know at the start, X has a higher concentration than Y. Immediately, A and B are gone. Then, now after 12 hours, what's going to happen? If my water molecules will move in and out, eventually it will reach this magical thing called equilibrium. Okay? So equilibrium means what? A and B are out. I know it cannot be less because it cannot keep going all the way out. I know C is going to be out. Answer here is just D. So you see the three-step process. I identify first, osmosis, diffusion, or active transport question. Once I identify it, I try to understand what are the different regions. Which one higher, which one lower. Okay. Third step, I draw the arrow. Once you do all three of these, the question becomes very straightforward. But for osmosis question, take note, are they asking you in terms of the concentration or the water potential? Okay. Then number 2, one beaker contains water, another contains a concentrated salt solution, red blood cell put into 2. Which diagram shows after 50 minutes what will happen? So concentrated salt solution, once again, this one, going to be osmosis or diffusion. Okay, osmosis or diffusion. What will cause the red blood cell to change shape? If my red blood cell change shape, is it diffusion of salt in or water molecules moving by osmosis? Step 1, osmosis. Then concentrated salt solution, water potential. Step 2, draw my arrow. Water will move out of my red blood cell. Just step 3. So concentrated salt solution, here is a lower water potential. Red blood cell is higher. Step 2, step 1. Step 3, I draw my arrow. So if water is going to leave my red blood cell, what's going to happen to the right side? It will shrink. So if I know it's going to shrink, straight away, A and B are gone. Then now left this one. Red blood cell and water. Now, I don't need to give you the water potential of water. Take note, in the exam, water potential of water is always 100% water. Nothing will have a higher water potential than water, whether it's distilled water or not. So you can assume, wow, this one, water is going to enter my red blood cell. Then what happens to my red blood cell? Will it become turgid like the plant or will it explode? So remember, ACL, animal cells will crenate or lyse, always chasing lights. So lyse is going to happen. So boom, it will disappear lah. So what's the answer here? The answer here will be C. This one, I have no idea what is this. Okay, this one is confirmed wrong. Okay, and all right. In lieu of time, let me go to a structured question. Okay, now part A, 5A, there are three ways in which substances move into and out of the cell of living organisms. Now, I chose this question here because I want to highlight this word here, compare. So a lot of you may not know this. Maybe you forget. Compare means I have to write two things. I have to write my similarities and my differences. Okay, similarities or differences. Both, I must have both. For compare, I must have both. And for 3 marks, what's the answer? I must have minimum 1. This one, also minimum 1. So it can be 2 similarities, 1 difference. 2 differences, 1 similarity also can. Okay, next, the way you compare this is what? Point-to-point comparison. So let's say, for example, I want to compare all 3. Okay, so active transport, osmosis, diffusion. Wow, how to compare? So let's compare similarity first. What is similar across 3 things? In my osmosis, in my diffusion, and here's what? Things need to move, right? When they need to move, there's always what? There is net movement of substances in all 3. That's one. Second one, all 3, okay, involve a water potential or concentration gradient. Or you can phrase it as, in all 3 processes, there are differences in concentration/water potential between different regions. Then what are the differences? Active transport, osmosis, need a cell surface membrane. Osmosis and transport need a cell surface membrane. Diffusion don't need. What's different between osmosis and diffusion? And difference between active transport, osmosis, and diffusion. Active transport requires energy. The other two don't need. Active transport is against the concentration gradient. The other two is along a concentration gradient. So notice I always compare point-to-point comparison. What do I mean? Active transport is against the concentration gradient. In diffusion and osmosis, it is down a concentration gradient. What is my point of comparison here? Concentration gradient. What would be wrong is to say, oh, active transport is against the concentration gradient, but osmosis and diffusion, okay, do not require a membrane, that kind of thing. One is about membrane, one is about concentration gradient. Two totally wrong things. Okay, so that's called not a consistent point of comparison. All right. This one, okay, so once again, okay, do a 3-step process. B and C were placed in sugar solutions of different concentrations. A is the OG, original. So in B, what happens? The fact that it is moving in, okay, what is moving in over here? Sugar solutions. So sugar solution, the diffusion or osmosis, I cannot tell, but think about it. The plant cell is getting bigger, the plant cell is getting smaller. So this is osmosis. So, osmosis. Then if it gets bigger, wow, water molecules are moving in. And therefore, I can do my region now. It means outside here is higher potential, higher water potential. This one over here, water is leaving outside. So it means here, it has lower water potential. Then from there, oh, I can find my concentration. In C, if I have a lower water potential, it means it's more concentrated. If there's a high water potential, it means it's less concentrated. Okay? Then you go about and explain the answer. Quite straightforward. Okay, so in lieu of time, I can't go through anymore. Okay, but what I will do here is this. I will show you the answers, which you all can take a screenshot and take down. So this is the one for, okay, hold on, come here. Okay. This is the answer, okay, for the cell signal, for the cell structure one. Okay, take a screenshot. I will also put it in the link later where you are downloading stuff. So don't worry, I'll put it there as well. Okay, but those who no time, you can screenshot this. This is the answers, okay, for my cell structure one. This is the answers for the membrane transport question over here. Okay, like that. All right. So you can screenshot it. If not, later I will put my answers in the drive. So don't worry too much about it lah. Okay. Now, last but not least, okay, I am going to go back to one last part about today. So, okay, share screen. So, indulge me for a bit. All right. So last part for today. Thank you so much for joining me. Okay. It will be really very great if you can give me feedback on how you found today's review session. All right. So just scan the QR code. Okay. And some not bluffing you top reviewer will receive a free set of notes worth $40. And you can check our reviews on our websites. We have been giving out a lot of free stuff. So most recently, one guy won a review, right? We will say we're giving out iPad. People don't believe. We gave out iPad. And that guy gave the iPad to his brother. Okay, so no cap. Go and give the review. Let me know how it's like. It can be whether good or bad. I don't mind as long as it's good review. As long as the review that I can use. Okay, so do scan this QR code and do the review. Okay. All right. Okay. And last but not least, okay, if you're interested to join me for classes, okay, I'm Tutor Glendon. Once again, I'm Tutor Glendon. My name is over here. Okay, Tutor Glendon. I have a regular Sec 3 bio class at Kovan on Sunday, 10 to 12 a.m. Okay, so if you would like to join me and see me for more lessons, you can also scan this QR code, okay, to indicate and find out more timings, or to say, hey, can I come for a trial lesson at Mr. Glendon? I want to find out more about his class. All right. If you can't make it for Kovan on Sunday, I have an equally qualified tutor, okay, Mr. Jason, okay, who also has a Tampines class on Tuesday. Okay. All right. If not, that's all from me. Okay. I enjoyed having you all around today. Okay. Thank you so much. All the best to your WA1s, all of you. All right. Once you're done with the survey, okay, you can just head off for your dinner lah. Okay. TGIF, go enjoy and all the best for your WA1. See you. Bye bye. Welcome, Avilia. Glad to have you today. Bye, Zena. See ya. Welcome. Thanks, Xander. Okay. See you. All the best for WA1. Okay. Annie and Belinda, you need more time? If not, I'm going to close the session. Are you all there? Okay, if not, wishing you all the best for WA1, Annie and Belinda. See ya. Bye bye.